presentation covers the importance of nutrition in the condition of the hive. For some background, the European honeybee, Apis mellifera, is a eusocial insect native to Europe, hence the name European honeybee. And being eusocial, they operate as a hierarchy with job assignments, including the queen, workers with several subcategories and subassignments in the worker category, and drones. And only queens can reproduce. They mate with multiple drones to store sperm in the spermatheca for increased fitness. And high quality queens are vital for colony quality. And they live the longest of any bee in the hive. And the workers all pour in resources to develop future queen larvae. Queen selection typically takes place during swarming when the queen is dying or when the queen is killed. And she usually develops in a specially built cell. As you can see in the picture, it's a large peanut shape. And she is usually flooded with royal jelly her entire larval stage. However, the unexpected can occur, which I'll discuss in a later slide. And when this happens, the workers have to begin to improvise by flooding normal worker cells, which are the smaller capped cells, with royal jelly. Some situations that include or that call for queen rearing includes reproduction through swarming, a failing queen, or if the queen is killed and needs to be replaced. My research question is, how do workers select a replacement for the queen when the queen is unable or unfit to decide? The first factor I found that impacts queen rearing decisions is nutrition quality. An experiment by Sigilli et al. Compared the number of queens produced from worker larvae supplied with adequate nutrition to hives deprived of food. With larvae in hives supplied with ample food. They found that the number of larvae selected to be queens was much higher in colonies with an abundance of nutrients. As you can see in the graph, the light gray represents the non-deprived larvae in the hives, and the dark gray represents nutritionally deprived larvae. And there was over double the number of queens reared to pupation when there was ample nutrition supply. Another factor that affects queen selection is found in their genome and affected by diet. The larval diet is critical to the function of enzymes, pheromones, and genes in larvae and adults. In this graph, you can see that the enzyme DNMT3, which is found in honey and bee bread, the usual worker diet, was much lower in queens fed royal jelly for 5 days versus for 3 days, indicating that the duration of royal jelly feeding affects enzymes present in later life stages. This time constraint is especially important when you consider the lack of time available to consume royal jelly in an emergency situation, such as the failing queen, because colonies don't rear queens to have on hand. So when the new queen emerges, the rest of the queen cells don't emerge. They aren't allowed to emerge. And if 2 queens emerge at once, they will fight to the death until the surviving queen becomes the new queen of that hive. So if a new queen is needed in a day or two, the larvae may not have enough time to consume royal jelly to become a high quality queen. And as I mentioned earlier, a high quality queen is essential to the success of the overall success of the hive. And as you can see, queens reared on royal jelly for 5 days had about half the DNMT3 activity as the queens fed royal jelly for 3 days, further illustrating that diet really impacts growth and development. And in the bottom graph, you can see that the longer royal jelly was supplied to all of the worker and queen cells, the more queens and less workers were produced. So by the end of day 5, it was 100% queens and no workers or intercaste. Physical characteristics are also very important for bees in all castes. Workers don't have the ability to mate or lay eggs, so her ovaries are smaller than the queen's ovaries. In the graph, we can see that larval diet is a driving factor in their ovary size, thus affecting queen worker differentiation. Mao et al. Added p-coumaric acid, which is again found in honey and bee bread, the normal worker diet, to the standard general diet of honeybees and measured the ovary size. And the larvae reared on a diet high in p-coumaric acid concluded that that showed reduction in ovary size compared to those reared on standard diet, illustrating that the diet of the worker is the reason for the lack in ovary size and function. The results of each study discussed point to clear results that workers who are choosing the replacement queen prefer well-fed larvae during emergency queen selection, as they can detect pheromones such as the hunger pheromone, and the number of queens produced increases when the reared larvae are in a nutritionally stable environment. Nutrition impacts the genes expressed as well, such as genes for ovary size and general queen development. In conclusion, a high quality diet increases the fitness of that larva, meaning larvae reared on a high quality diet will be selected as queen, since biology is generally driven by fitness. And the worker bees decide which larvae will be reared as queens based on emitted pheromones, which are affected by diet. And contrary to previous belief that the chosen queen has genetic relation to the worker bees in the previous um, generations. That is actually not the case. Emergency queen selection is not affected by a genetic relation to worker bees. It is completely affected by the quality of nutrition that the chosen larvae is reared on. This knowledge is significant because the queen affects the entire hive. If the queen isn't thriving, the workers may invest more time, energy, and resources into helping the queen just survive or rearing a new queen instead of benefiting the future of the hive during their normal tasks, such as foraging, scouting, rearing more larvae, making bee bread, and processing honey, etc. This impacts beekeepers since an abundance of foraging resources in their area ensures a high quality hive function.